Alright, so I just spent a bunch of time making a video, about 15 minutes, and it's not working. So this is part two redone. So I might have to apologize if I skip some things or if I forget to mention some things, because I've just done this here on a snow day. Not that I don't like being here. I actually do enjoy being here, getting work done, but don't like doing things twice. Hate doing things twice. So just gonna, use another 15 minutes of my life explaining this the second time. Hopefully it's pretty good. We've just talked about cell cycle and checkpoints and enzymes that are involved and reactions that are catalyzed that allow cells to realize if they are where they need to be, if everything's been done properly, and if not, those checkpoints allow the cell to stop dividing so that a cancerous cell or an uncontrollably dividing cell doesn't occur. All right. So these cell cycle checkpoints are very important. All right. All this stuff, the CDKs, all right, the cyclins, the cyclin-dependent kinase enzyme substrate complexes with the cyclins, the phosphorylations going on, the checkpoints all going on. Very important. The synthesis and degradation of each different type of cyclin. All right. Being synthesized, being degraded. Being synthesized, degraded. Synthesized, degraded. All that stuff is really, really important to how cells function normally. And if something doesn't, if all of these things are being made by the DNA, right? So if your DNA isn't replicated properly, you don't make the right things. And if you don't make the right things, you're going to have cells that are going to be out of whack. Okay, and this can happen spontaneously. It doesn't have to be. There doesn't even have to be a reason for mutations to occur that cause cancer cells. Oftentimes there are reasons, but there doesn't have to be a reason. These things happen. There are problems that occur during replication of DNA that are completely natural. Oftentimes these problems have resulted in novel adaptations for organisms. So replication of DNA not being picture perfect is actually a way in which variation has increased, and we all know that variation in the environment is good because it allows organisms to adapt to changing conditions if the conditions force them to do so. So we are now talking about mitosis. Remember, mitosis is a form of cell division for somatic cells or body cells. We're going to stick to talking about multicellular organisms right now, because single-celled organisms often divide in different ways. Like bacteria use a process called binary fission, which is very similar to mitosis, but not exactly the same. But we'll talk about that when we talk about more about bacteria. These cells result in identical offspring. You don't want to be creating cells that are important in muscles in the shin or Achilles tendon, muscles that give rise to muscle cells in your Achilles tendon. You don't want them giving rise to cells that are designed to be functioning in a bicep or a tricep or an eye or a hair follicle. So we want to create cells that are identical to the cells before them. All right. So we want cells of our skin, certain areas of our skin, like in our hand, to be giving rise to new cells that are serving the same purpose. Remember, every cell in your body has the same DNA, and when it replicates all of its DNA, except only some of that DNA is actually being expressed or used, and one cell passes on that same information. Yes, I'm replicating all my DNA, but I'm expressing only specific parts of that DNA, and that's why this type of cell division is so important. Because what's going on here are there certain parts of your DNA here that are actually not being expressed, they're being silenced, all right, by certain proteins, or they're being silenced by specific functional groups, all right, like methyl groups, CH3, are often in DNA regions that are being silenced or not expressed, or actually have a lot of what we call methylation. So we want to pass on those same properties to the next cell, right? So one cell splits into two. We have a parent cell, which gives rise to two daughter cells. The process includes interphase and M phase, or the mitotic phase. Okay? First thing we need to talk about was a little about interphase. Interphase, the cell spends most of its time, at around 80 to 95% of its time in interphase. It spends most of its interphase time in the growth phase, G1. G1 is where a cell is normally functioning. All right, so most of the cells in your body right now are functioning normally in the G1 of the interphase. All right, but once a signal comes either internally from the cell or externally from things like growth hormone, growth hormones are signals that are sent from certain regions of your brain that are designed to tell other cells to grow and to divide. All right, it's one of the reasons why people that take growth hormone are also able to produce muscles that are so much bigger because they can produce more muscle cells. All right, now this has extremely huge detrimental effects on other parts of the body and the brain, and the future of things like aging and things like that, and hormone balance. But some people believe that they're willing to take those risks in order to be superior at specific parts of their life, like sports, for example. But in the end, I think everyone ultimately finds out that it's not worth to be taking more growth hormone than already naturally occurs in the body. Anyway, interphase occurs in G1, S, and G2. Most of the time is spent in the growth phase. You'll have certain cyclin CDK complexes that will be synthesized here to keep the cell in G1. And once those get degraded, the degradation sets the signal for more cyclins and CDK complexes to tell the cell to enter S phase. In S phase, the DNA here, which occurs as chromatin in the nucleus, this is the nuclear envelope. Chromatin is an unwound, uncondensed form of DNA. All right, and it's easy to replicate DNA in the chromatin form. All right, although don't look at these pictures, just please take my word for it first. I'll try to show you some other pictures. You can't really see chromatin under a microscope, but you can often see the compact form of DNA as the chromosome. Chromosome is a compact form of DNA, chromatin is not. So the S phase, you're duplicating your DNA, all of it. All right, so all of your DNA is duplicated. And as we enter G2, we're going to start synthesizing, duplicating other things like organelles. We're going to start producing lots of lipids and phospholipids and enzymes that are going to be necessary for the enzymes that are going to be necessary for entering each phase here. Enzymes that are going to be necessary to make protein. As you're going to see, proteins are probably one of the most important things about cell division. All right, and once we are done with G2, we've officially entered the M phase, or the mitosis phase. All right, mitosis includes prophase, metaphase, anaphase, and telophase. Cytokinesis is a process that occurs in telophases, but it's really not its own phase. All right. Please feel free to write down any questions you have. I'd love to answer them in class. From interphase, we've now decided that everything's gone right. We're growing fine. We've replicated our DNA good. We're starting to produce all the things that are necessary for mitosis, and we're saying, okay, we are ready to go. There's no problems. If there were issues here, some signal would be sent, and that signal would tell the cell to stop dividing, don't continue, and it will either self-destruct or try to fix what went wrong before entering the M phase. All right. The M phase, also known as the mitosis phase. Remember, we're producing identical offspring here. We're trying to produce identical offspring. All right. So in interphase, our DNA exists as chromatin. Our nuclear envelope exists. We have these two things called centrosomes. A centrosome is a place where a centriole exists. All right. Now centrioles are these two little structures here that are microtubule organizing centers. They're the place where microtubules will be synthesized. As you're going to see, microtubules are probably one of the most important structural components of cell division. All right. So we decided to enter the M phase. We've duplicated our DNA. These centrosomes begin to form, or the centrioles, the centrioles, which are the structures, and the centrosomes, which are the places, begin to form at one end. And as we enter the prophase of mitosis, now we've entered the actual dividing phase. Our centrosomes begin to move towards the poles, and our centrioles are starting to synthesize microtubules, and you can see these microtubules are beginning to extend. All right. Now, we're going to treat prophase as its own phase, and prometaphase we're going to meld this into prophase. All right. I don't like the idea of prometaphase. It's not necessary really. Think of prometaphase right now as metaphase, as prophase. Think of prometaphase as prophase. So in this huge phase, what's happening? Our chromatin is condensing to chromosomes, really condensed form of DNA. Our nuclear envelope is beginning to break down into pieces. All right? Our centrosomes are moving towards the poles and actually almost get to the poles at this point during prophase. Our chromosomes are beginning to try and line up at the center. All right? So we have a nuclear envelope breaking down. We have our centrosomes at the poles. Our centrioles are making microtubules. This whole thing is called the mitotic spindle. All right? Mitotic spindle is starting to make microtubules that are extending towards these chromosomes here. And these are identical copies. Each side of this, all right, each side of the X is an identical of the other side. All right? So these are called sister chromatids. All right? So we have nuclear envelope, centrioles, the region is centrosome. They become the mitotic spindles. All right? We have chromatin, and now we have our condensed form, which is called the chromosome, and attached to an identical copy, we call them sister chromatids. All right? They are attached at the center by something called a centromere, which is a protein that keeps them together. All right, at the centromere. So now, at the end of prophase, we have our nuclear envelope totally broken down. Our spindles are now near the poles or opposite ends. Our sister chromatids are beginning to line up at the center. All right. Now we enter metaphase. And metaphase is described as the time or place by which our sister chromatids are completely lined up at the center. It's called the metaphase plate. All right. The metaphase plate is the place where these chromosomes are going to begin to separate from one another, these sister chromatids. Now at each end, the mitotic spindle, as you can see, has extended these long microtubules. Some of the microtubules are going to actually attach to the centromere region of the sister chromatids, and as they attach, we're going to enter a next phase called anaphase, where they begin to degrade the microtubules and start to pull. All right. We're going to start to pull, our sister chromatids apart. All right. At this point, what they're not really showing is that the cell is actually beginning to elongate. All right. Let's see. Show you better. Anaphase. Yeah, this is a better picture here. As you can see, during anaphase, the cell is beginning to pull apart from one another. All right, so the cell is beginning to elongate. They didn't really show that before. Okay, it's really important. The cell is beginning to elongate. This is that's meiosis. Sorry, this is all meiosis. Anaphase. Mitosis. There we go. The cell is beginning to pull apart, and the pulling, actually, the cell becoming longer, actually is a product of the spin of the microtubules pushing and pulling cytoplasm and cytosol. The microtubules are actually pushing and pulling cytosol apart from one another, and the cell membrane is beginning to elongate or pull apart. And remember, if we're creating two new cells, we need double everything. We need double nutrients. We need double the organelles. We need double the phospholipid bilayer. We need double the proteins. We need double everything. So this process is intense. The cell's going through like a seriously intense process right now. All right. So now we're in anaphase. Our sister chromatids are now pulled apart. They're called daughter chromosomes. and we enter something called telophase. At telophase, you see this right here. This is called the cleavage furrow. All right, the cleavage furrow is the place by which the cell membrane begins to pinch in, and new cell membrane is beginning to be synthesized in the center. All right. Now, this is an animal cell. If this was a plant cell, something would form in the center called the cell plate. Images. All right. What we're showing here is these two cells have just gone division. There's a cell membrane and a cell wall here. But what's forming here is a big band of cellulose, which is going to form the cell wall. All right. This is called the cell plate right now. The cell plate is forming, it's cellulose being synthesized, and it's also going to form the cell wall that separates these two cells. So in plant cells, a cell plate is formed. Okay. Let's see here. Okay. Now, the process of the actual dividing of the cytoplasm and the formation of the cell membrane or the cell plate is called cytokinesis. Cytokinesis is a process of cytoplasmic splitting that occurs in telophase. All right. As you can see, the nuclear envelope begins to reform, the mitotic spindle begins to degrade, and what's going to happen is the DNA is going to start to unravel and become chromatin again, and both of these new cells are going to enter G1 of interphase, where they will spend a majority of their time until they need or are directed to split again.